Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is the RAS MAP kinase pathway. Okay, and before going to this topic, I want to say that about the receptor tyrosine kinases I have already uploaded in my previous video, so you just check out that one. And I thank you to all of you who have already subscribed my channel. And I say to it is my humble request to the newcomers, please you share and subscribe this channel if this is helpful to you, and continue with my channel. Okay, so let's continue the topic. What is this RAS MAP kinase pathway? This is the pathway that is by RTKs, that means receptor tyrosine kinases. It has three pathways. So what are the another two pathways? I try to describe after this. So first pathway, that is the RAS MAP kinase pathway. So RAS is what? RAS is an intracellular monomeric GTPase switch protein, and that helps in the signal transduction. Okay. So actually some ligand induced activation of RTK that stimulates the tyrosine kinase activity. First, when the ligand bind to the RTKs, that means the receptor tyrosine kinases, they become activated and they stimulate the tyrosine kinase activity, which stimulates several signal transduction pathways. So this RAS MAP kinase pathway is one kind of signal transduction pathway, and here the RAS is one kind of protein. Okay. So here generally three closely related RAS proteins, these are seen in the human body, and RAS is one kind of protein where the lipids are linked covalently. Okay, and they present in the cytoplasmic phase of the membrane, and is actually RAS cycles is continuing from where? From the inactive GD bound form to active GTP bound form. Again, RAS cycles that continuing between inactive GDP bound form and active GTP bound form. Okay, this requires as another assistance of two proteins, GEF and GAP. Two proteins are very important that generally help in the RAS cycling process. Okay, and the GEF, full form of the GEF is guanine nucleotide exchange factor, and GAP means GTPase activating protein. Okay, so RAS is one kind of protein, that is the obviously what kind of protein? I am saying that this is the GTPase switch protein, but for continuing the process there are another two proteins very important and that helpful to this RAS protein. These are the GEF protein and GAP protein. Clear? Now how the RAS works? The RAS activation is accelerated by GEF, which binds to the RAS GDP complex. Okay, so first of all, RAS protein that binds with the GDP, and this GEF protein also binds with the RAS GDP complex. Why they bind? Generally, this when GEF binds to the RAS GDP complex, this causing the dissociation of the bound GDP with the release of GEF. That means when GEF is bound to this complex, it helps to dissociate this RAS from GDP, and after that GEF also released from this complex. Clear? Again I repeat, when GEF that bound to the RAS GDP, then it helps to dissociate this RAS from GDP. Okay, and again GEF also released from the complex. Okay, and what is the role of the gap? Gap generally binds to RAS GTP and accelerates its intrinsic GTPase activity. Okay. So what is the role of GAP? GAP generally binds with the RAS GTP complex and it accelerates GTPase activity. So two functions are very different, the role of GEF and the role of GAP. Generally GEF that binds with the RAS GDP and GAP that binds with the RAS GDP. So when GEF bound to the RAS GDP, that dissociates bound GDP and also release GEF own or itself. And when GAP that binds to the RAS GDP, it accelerates the GTPase activity. Clear? So binding of ligand to an RTK leads to activation of the RAS. Okay, so ligands has important role, that if ligands bind to RTK, that means the activity or the activation of the RAS that become increased. And there are two another proteins, one is the adapter, another is the GEF, that provide key link with the RTK and the RAS. Clear? So actually ligands have a very important role, because when ligands bind to the RTK, then activity of the RAS is found, and the interaction between the RAS and RTK is possible by the presence of two proteins. One is the adapter protein, another is the GEF protein. Clear? Next, what we will learn about the adapter protein. So adapter protein, what is saying that this binds to an activated receptor and then other proteins binds to them. What is saying that when adapter proteins, they bind to the activated receptor, that means ligand binded receptor or ligand bound receptor. Okay, and after just binding with the ligand bound receptor, what is the role of the adapter? They try to bring another proteins to bind with the activated receptor. Okay, because adapter has no role in catalytic activity of its own, but it helps to bring other proteins to the receptor. Okay, or helps to bind, you can say, other proteins towards the activated receptor. Clear? And this adapter protein, that is the example is GRB2. This binds to the specific phosphotyrosine on an activated ligand bound receptor and to the cytosolic GEF SOS protein. So what is saying that this is an example that GRB2 is one kind of adapter protein. When it binds with the receptor or ligand bound receptor, it attracts two kinds of protein. One is the GEF, another is the SOS protein to bind with the ligand bound receptor. Okay, and SOS then promotes the formation of RAS GTP, active RAS GTP. Okay, what is the role of the SOS protein? Its role is that it helps to promote the formation of active RAS GTP. Okay, and here another protein, this is the SRC homology domain, SH2. In GRB2 that binds to the specific phosphotyrosine residue in activated receptor. Okay, so SH2 is the SRC homology domain. So this in GRB2 binds to the specific phosphotyrosine residue, and in activated receptor they also bind to the activated receptor. And GRB2 has SH2 that activate SOS. Okay, there is the role of SH2 is that it activates SOS protein and when SOS protein becomes active, then they promote the formation of the RAS GTP. Clear? That means there is important function of SH2, because if SH2 is not there with the GRB2, then it's not possible the activation of the SOS protein. Okay, and if the activation of SOS protein is not occurring, that means the active RAS GTP formation is not possible. Clear? So, and there is important thing is that this SH2 domain is first identified from one viral sequence or one virus, you can say. This is the oncogene virus, obviously. This is the VSRC. Okay. So this is the actual topic, or the activation of the RAS MAP kinase pathway. So here important thing is that GEF has important role, okay, and GAP has important role, and another important role is C, adapter protein. Okay, because you have to remind that GEF actually binds with the RAS GTP for what? If this try to bind with this, that means dissociate the complex or dissociate the bond that is formed with the RAS GTP. Okay, but when gap is bound with this RAS GTP, that means it accelerates the GTPase activity. Clear? And adapter protein has important role is that this adapter protein has actually not important role in the catalytic activity, but important role is that it try to bring another protein to bind with the activated receptor, and these proteins have important role like that GRB2, or you can say SOS protein. These are very important because GRB2 why important? If GRB2 is added with the activated receptor, it contains SH2. Okay, and what is the role of the SH2? This actually activates SOS protein. Clear? This SOS protein activates the actually RAS GTP. Okay, that means different kinds of proteins, they are linked with each other for the activation of any RAS proteins, or you can say to create functions or to induce the RAS protein to continue the phosphorylation process. Okay, so here another important thing is that the ligand. If ligand is not bound to the receptor, so receptor is not activated. So ligand obviously or must have to bind with the receptor for getting you just say that try to activate the receptors, and if receptor is not activated, that means no proteins bind. If the proteins bind with the activated receptor, there is no signal transduction occurring because the receptor is not activated. Okay, that means first receptors have to active, then proteins bind with that, they active and continue the transduction process. Clear? So this is the RAS MAP kinase pathway. Another important pathway is that only MAP kinase pathway. So this is so large, so I cannot complete the total topic in one video, so it's not possible to me, and that's why here I only try to describe about the RAS MAP kinase pathway.